So we are starting recording now. Welcome everybody to the meeting number 4. We have almost 60 slides today and many different presenters. The slides, I will just share my screen here, are available. You can also get to the link via our ASCT Slack channel. And they are also here in the chat window. You see the first slide, and I left in the key takeaways from meeting number 1, 2, and 3. I will not go over them because we have very limited time today, but you might also have taken away other items, but these are major items that we discussed in these meetings. The agenda for today was shared. We will have a quick work organization on my end. Then I added a section on recent publications. There's a lot of good work that is getting published right now, also data sets becoming available. And I would love you all to help us understand what else you see that's relevant for the ASCT table effort. Sarah has been sending me a lot of good pointers, so I included some of them here. Then we go over to presentations and discussions of new ASCT tables. We have skin, spleen and lymph nodes by Mara, and then brain with Amy Bernard, and a little bit of on the lung. There's a team grouping and evolving, and I think we absolutely, with the pandemic going on, want an ASCT table for the lung. Then we have presentations on the cell types and ontologies, and one is by Anna Maria on cell ontology usage. One is by the broad team on standardization of gene names and genome builds. And then there's one from our team, MCIU, by Ellen on flattened ASCT+B tables and biomarker issues. And then if there's time left, I'm happy to talk more about how we are using these ASCT+B tables to make them machine readable and also still keeping them human readable. So that's really a big challenge. So going over to the work organization, we have a roll call sheet, and I hope you can all click on the link and add an X on the respective column, which is the K column, like my first name. So please indicate if you are attending or not. This way we also know who is active when and who has heard what, even though everything is recorded and shared back, and the slides are also freely available. Second, we now have an online form which you and others can use to register for this effort. This way we can make sure that everybody gets an invitation to these meetings. We also then have a little bit more information on the expertise that people bring to the table, if they're coming from the pathology or anatomy or surgery side, if they are from academia, industry, or government. I think it's very, very important to understand who is participating in these discussions so that we can use your expertise most wisely. Many of you have been active on the Slack channel. We would love to increase activity, and I think we will get there as soon as we add more programmers which can use these flattened tables in a much more programmatic way. But I believe even I and many of you can use Slack efficiently, and it actually sends out emails also. So it's a very efficient way to group us into organs, even though we wanted to break down these silos, but I think the discussions are still very much focused on specific ASCT+B tables, even though we start to see commonalities and differences and cross-cutting challenges, definitely. But then also please do use the general channel and also the publications, tools, data sites channel to help us understand what other papers are getting published, what data becomes available, what events are coming up. For instance, our team will organize a cell type identification hackathon on September 23rd. So as we have more information becoming available, we will post it there. But you might have other events that with COVID might be virtual, so we can all more easily attend them. So please use that as a way to organize communication so that it doesn't get lost in emails. Slack is also wonderful in that people which come in later, they still get to see all the previous communication. So it's they can very quickly catch up to the state of our understanding. I added another slide here because there might be positions that are specific to ASCT+B tables. And I think Griffin is trying to hire the best postdoc he can get. And we have the position available right now. Griffin, do you want to tell a little bit more about this position?
Yeah, so this is someone to help us focus on building out the network of the vasculature, all the way from major arteries and veins down to patterns of the microvasculature. It's not so much that HubMap is interested in a model of the vascular system, but it's that vasculature passes through everybody's ASCT tables. We want to use the vasculature as kind of a coordinate system to identify location in the body. A lot of this position is a literature search. There's a lot of separate studies that have been done for different organs and different species, mapping out the microvasculature, the whole tree within a particular organ. There's also different nomenclatures on how people describe the microvasculature, things like branching depths and vessel diameters and lengths. So figuring out what are all the parts of the body where the vasculature has been well documented and defined, and what are the labs that are currently working on this, and what are the different techniques people are doing. Neither Caddy nor Hire, really, this is our area. We're not really sure where the type of person who would be good at this would be looking for postdoc positions. So we probably are looking for someone with a PhD in anatomy or cell biology, radiology, pathology, but we would like some advice on if any of you have some students or trainees looking for positions or know where we might go to find someone.
Thank you, Griffin. And I think over time, we might also use these postdocs to pollinate across the different labs. I think the best way of packaging up a piece of knowledge is in a person and then sending that person off to another place. So I think over time, we will see more and more summer internships happening around this effort, or sabbaticals or postdocs. So please also post in the future your open positions here, or just send them to me and I add them to the slides. I think it's very important that we do not just share data and publications, but also people. I think this will lead to much more meaningful collaborations. Going back to the agenda, I wanted to make sure that you all saw three papers that I discovered, and you might have discovered even more relevant ones. So please do post them via Slack or make them otherwise available to me, to us. One of them is, Ellen pointed me to the cell marker, and I don't know how widely that data is used. There are a number of other similar sites out there, and basically people manually went through much literature, here in this case over 100,000 published papers, and they identified 13,000 cell markers of 467 cell types in 158 human tissues. And again, this is not the only effort, there are others out there. And I'm very curious to understand what teams here on the call are using of this, if they are using other efforts like it, because ideally, it's not 10 teams which all read those 100,000 papers, but it's just a more communal effort. And I think we will need some of this to be done, for instance, for the microvasculature, because I don't think it exists. So that was the postdoc position that Griffin just introduced to you. But we should benefit from other efforts like this. Maybe I stop here for a moment and see if other people have comments on this particular effort. Has anyone used it?
I have not used it. So what's, I mean, I assume that there is information about the quality of the databases of these annotations and things like that, right?
They have links back to the papers that claimed that there are certain markers for certain cell types. And I think that's ultimately the type of provenance that we really would love to have for the ASCT tables. So feel free to also comment to this on this in the chat log. Again, I think it's really important for us all to understand what other resources exist.
I can comment a little bit. I've used it. A lot of the studies tend to be bulk RNA-seq studies. Mm-hmm. So then sometimes the marker genes might have shifted a little bit, because in a bulk RNA-seq study, you often sort a cell subset down to a very fine level and then compare it to another similar cell subset. So when we're doing single-cell RNA-seq, we tend to have a more heterogeneous population. So some of the marker genes may not apply, but it usually does out kind of the major ones. So like major cell subsets are pretty easily defined here. but once you try to take a subtype, it's harder.
Okay. And the nice thing is that this particular source actually makes it easy to download the data. And then you could compare it with other data sets that have been constructed manually or very soon also more automatically. We had a great discussion on Wednesday with Rahul on how to keep provenance when initially it's mostly human decision-making, but very soon it will be algorithm decision-making 80% of the time probably. And so it's very, very important to keep provenance for where certain cell type identifications come from. And again, existing resources and pointers to those would be very valuable. All right, in the interest of time, as we have almost 60 slides today, there's another paper which just came out, which has a large table of cell types for the human lung. And it's organized in a slightly different way. It's organized by cell types, but still very, very interesting because it connects cell types with the canonical markers. It also has the relative abundance and the number in millions, extremely valuable information on the CCF side. So if those authors and reviewers of existing ASCT tables could also help us all understand if that information is already available and how we could get to it, that would be very valuable going forward. You also have in this particular work links to relevant papers. So again, it's excellent to see those citation references. And again, a nudge to those which design and also the ASCT tables to keep track of provenance. So if there are papers that report the usage of certain markers for certain cell types, please do provide that information. Try to get it into that table. I know right now this might be not easy, but I think you can find a way or just get back to us with different suggestions. But going forward, I think we want to keep track of who said what, be it a human person or an algorithm. Another paper that I found extremely valuable is one on the atlas of a rat heart in this case. And there's a similar effort now going on for human. And again, here you have a number of areas identified and also explicit, specific and spatially explicit transcriptional gradients. And so for the CCF design, again, I would love to have more pointers to papers that help us all understand what anatomically specific and spatially explicit transcriptional gradients exist for which cell types. So if you see papers on this, please post them in the Slack channel or send them to me or Ellen directly. We would love to hear from you. And I think there will be specific sections of this particular call that will just try to bundle some of this work and help us all understand what is known and what is not yet known. All right, presentations and discussions of new ASCT+B tables. Skin is first, so Anup Sood and his team, they are going to present on the skin. Anup, are you ready for this?
Yep.
All right.
Thanks, Gary. So this table was put together with three experts, Thomas Brand, Jonathan Ho, and Joel Sunshine. I don't know if any one of them is on the call today, but if you are, please speak up if I mess up things.
Okay. Anup, I'm on. So
pardon?
Anup, I'm on, Joel. So I'm here if there's any questions. Sure,
thanks. So skin is a very large organ, and there is a lot of variation from region to region in the body. So what we have tried to do right now is just look at a very high level. What are the major layers or structures present in the skin, and what are the different cell types within those structures or layers? The variability that exists from side to side is more related to the thickness of these layers or structures, or the number of these structures or different cell types that may be present. In some cases, some of the structures may be absent altogether. So in the initial effort, if you could just go to the next table. So at the high level, so there are three major layers in the skin: epidermis, dermis, and subcutaneous. And then within these layers, especially in the epidermis, you have multiple different cell types. There are multiple layers of different cell types. And then there are several structures in the epidermis that actually dip into the dermis. So you may see some of these epidermal structures like hair follicles that are actually, they look like they are present in the dermis. And then in the next column, we have kind of highlighted what are the different cell types that are present in these layers. So in case of epidermis, there are markers for these individual layers of cells that can essentially differentiate some of these layers. In the dermis, there are two major layers, but they are essentially composed of the same set of markers. And the cell types that are present in those are also the same cell types, they're just distributed differently. And I think what we're planning to do in the future is actually dig deep into individual regions where there might be big differences in the skin composition, like eyelids or scalp or bottom of the foot. So we will create separate smaller tables for those regions. And I think the challenges in this case are probably similar to the challenges everybody has. How do you find unique markers for individual cell types? Because a lot of these markers end up being expressed in multiple cell types, even though you may see higher expression in one cell, they're likely present in some other cell types. That we will, in each case, we will need multiple markers to identify individual cells. So that's pretty much what I have to say. If there are any questions.
Thank you so very much, Anup.
Problem.
Now, I just had one quick question. It doesn't look like you have any stem cells on that list. Is it because the markers aren't known, or?
I don't know if there are specific markers. No, you're right, there stem cells are not listed. So we will certainly add that to it. Geology, do you know any markers, specific markers that are skin stem cell specific?
So I guess it depends on what we're talking about. So like, for example, I know that people have done a lot of work sort of molecularly trying to define, for example, hair follicle stem cells. We haven't, in this table, for example, we haven't really, dived down yet at this point into, say, to delineate in more detail sort of the hair follicle apparatus and the various components and some of the sort of the cycling markers. Some of those things we could work on that. In terms of other things, a lot of these other sets of cells in the dermis, they're not, they're going to be, mostly differentiated cells, and rather than stem cells that would be present. So, I think mostly what I think about when I think about stem cells in the epidermis is that involve that there's some in the hair follicle. There are some potential markers we could try to look into for other members of the epi. Would be the first thing to do, probably.
Thanks.
Thank you. In the interest of time, let's move on to the next organ, which is spleen. And I think Marta is going to explain the slides here.
Hi, guys. Basically, the point that we're at on our table right now is that our design decisions were based on what was called for our annotation portion of our HubMap project. So it is heavily focused on positive markers, and also it is something that we have kind of tried to evolve into based on what we are currently using in the lab. So therefore we could identify particular cell types. This is the existing chart as it is for the spleen. We started off with the major structure, went to substructures, went to tissues, to cell types, and to the marker subset, which I've already self-criticized as being too centric on what was called for our existing project. Okay, next slide. So Rahul did a set of analysis on the 10x data that came out of our spleen sets for HubMap. Through this, he identified 14 cell types. Currently, 8 of those cell types directly align with what we have in the ACS table. Of the remaining 7, several of those, it's just that we're breaking them down differently than he is. So he has more subsets of T cells than we are currently able to identify ourselves. He doesn't break his B cells down, whereas we do. So there's things like that that can be approached from both our side and his side to perhaps make these tables and the harmonization data line up a little bit better. Okay, next. And I put this here because I wanted to discuss the approach that we're taking where we're trying to look at vascularization for mapping. The spleen's a little complex on this because you have a situation where once the blood moves through the artery and the arterioles and down the peninsular arterioles, you have both an open circulation and a closed circulation. So basically what's happening is you have a portion of the blood that's basically pouring over an open complex of macrophages and then moving back into the sinuses. So it may be a little complex with the spleen when you get to trying to rely entirely on vasculature as a means for mapping in parts of the red pulp. Okay, next. So our challenges. The lymphatic vasculature isn't a great mapping approach either, at least in the spleen, because there's no afferent lymphatic vessels. And what is there is not located in the parenchyma. It's in the trabecula and the capsule and the hilum. So that was what I originally looked at, is could we get around the vasculature by following the lymphatic vessels? But it really looks like that is probably also not exactly the way to go with the spleen. And I just put a comment here trying to explain that basically the red pulp of the spleen within the cords, that's the only place in the entire human body that has an open circulation. Okay, next. And then we have a pretty long to-do list going here. We have just replaced the pathologist that we were using to read out our tissue with an actual hematopathologist. So I want him to hopefully review the table in the short term. We really need to recruit additional experts to help with editing. I don't consider myself an expert in the spleen. Basically what you have here is come from a pretty exhaustive literature search. So it would be great to be able to pull some more people in. I have my eyes on somebody that I may have Caddy maybe help me with, because she's located in Germany at a fairly small university. And apparently when they were first setting up the HubMap grant, they reached out to her to try to get her involved, and she wasn't really too hot on being a member of HubMap, but this might be a project that's more up her alley because she's done so much in the way of mapping structures within the spleen and understanding the cell types of the spleen. Then going on, we need to add marker genes to our table. Currently, everything's in terms of the antibodies that I'm using in multiplex staining. I really haven't done a lot to incorporate the available genetic markers. And then we need to probably go back to the table and actually search hard for these, I guess you're calling them the calling card, cell type specific markers, And I need to add the negative antibody expression where it's actually relevant to being able to identify cell types. And then an option I came up with for the spleen is also to maybe look at using reticulin framework as a possibility of something to map off of. It's probably going to be pretty different from spleen to spleen, but it would definitely give you a framework to work within in the spleen, since it's basically providing the entire framework that all of the cells sit in this sort of bag of immune cells that make up the spleen. And then last of all would be, of course, like I've already mentioned, to focus some more on trying to work with the proteomics data to harmonize between what we're putting in the table and what they're able to pick out. So that's pretty much where we are with the spleen.
Thank you, Marta. And I think you're also going on to lymph nodes. Is this the
I am. And a lot of the same issues, of course.
But let's stop for a moment and see if there are questions, comments.
I have a question. This is Zarina Gallus. So I understand what you are saying, that it's hard to map a pool, but do we need to map a pool? It's like trying to map the ocean. So all you can do usually is to map the edges, not the content of the ocean. Because if I remember my pathology well, the spleen is basically a sieve. So you pass stuff through it, and even the endothelium is large enough so you can have full cells passing through and so forth. So do we need to map pools, or we just need to map stationary structures and mark the edges of things that are in flux? Because if we need to map the pool, then we need to map the blood everywhere in the body, and I don't think that's possible.
And that is a great point, because, you know, there's so much blood moving through the spleen at any given moment. And I didn't really focus on trying to look for, say, granulocytes, because that shouldn't be part of the residential population of cells making up the actual spleen. That should be pretty much a passer through cell type. And I'm assuming that as we're trying to map, we're much more interested in the actual resident cells of an organ.
Yeah, correct. That's what I was thinking. So I think it may be interesting at some point, but I don't think we are there yet to map things that are in flux, going in and out, and not really resident.
I think it's similar. It could be similar in the heart. You're not looking at the blood flowing through the heart. You're looking at the cardiac muscles surrounding it.
Correct. You have big bags of blood in the heart that we are not going to map.
In the interest of time, because we have so many slides today, let's go on to the lymph. Marta, are you ready for doing that?
I am. So once again, the design was based off of what we were needing for HubMap, so it has the same exact issues that the spleen had. The biggest thing that comes into play with the lymph node is the challenges that you're going to get from size variation, and the fact that each lymph node kind of has its own repertoire going on based on what it's been exposed to. So variation is going to be incredibly high from one lymph node to another. Okay. And that's the table as it exists. It's pretty well following the same format of what we did for the spleen, of having structure and substructure and moving into cell types and marker subsets. And it has the same shortcomings in that it does not yet have the proteomic based gene data incorporated into it. And I did start in with putting more of the negative markers in this one though. So it's a little, probably actually a little further along than the spleen is in that point. Next. And that's just showing the vasculature and the lymphatic vasculature of the lymph node. And in this case, this is going to be probably an excellent tissue to map off of either or both of those structures. Next. Back to the table review by our newly added hematopathologist, trying to recruit some additional experts. I think right now HCA and our group at HubMap are the only ones that are looking at lymph node off of the different groups that have been identified as consortiums. So it's probably going to take some reach out to to find some people that would be willing to get involved in this. And once again, we'll be adding marker genes, cell type specific antibodies, and negative antibody expression information where it's relative to cell type designation. And I think that's it for me.
Thank you so very much, Gloria. Let's see if there are questions, comments on the lymph nodes. And also, if you know of experts or have husband and wives or friends that might be interested in serving as authors or reviewers, please do use the chat channel to communicate names, and also just institutions that have major experts. I'm happy to reach out.
So Kati, is there like a sort of explanation, short explanation? I know a lot of lymphatic experts, but what would I ask them to do? So some sort of call for action. If you give that to me, I can send to a lot of people who are getting funded by the NIH to do lymphatic research.
Oh, this is
what am I asking? What am I asking them to do?
So that's a to-do item for me, to just write up what this effort is all about and why we need these experts to provide their input to those table designs and reviews. So I will write this up.
So include the type of expertise you want and the type of commitment and stuff like that. You know, people are very busy. Just yesterday, I was talking to a surgeon who is removing lymph nodes. I'm pretty sure he knows a lot about it, but what I'm asking him to do, and does he have the time and all that? So
yeah, great. Thank you for suggesting that. I will write that up. Miss Gloria. And there are also now comments in the chat window. Any comments, questions, quick ones for Gloria and lymph nodes?
Sorry, that Katie, that was Marta, I think. That was
Marta, I'm so sorry.
That's okay. I'm sure Marta can answer the questions about lymph nodes better than I can.
All right, then let's move on to the brain. We have with us Amy Barnard, and it's a pleasure to have you here. As you know, Ellen has done much work on the brain, and I let her speak for the brain and the table there.
Hi, everybody. So as I think many of you might be aware, the brain isn't actually part of HubMap per se, but it's obviously part of the body, and it has cells, and people care deeply about it. So it's sort of here is this update that's just related to some of the work that we've done at the Allen Institute, and frankly, just more of an informed that spans out to some very, very large efforts going on through an entire network funded by the NIH with the NIH Brain Initiative, that includes the Cell Census Network. So there's a whole working group on this, and I don't mean to capture it here. I'm just really reflecting what's coming from the Allen Institute for Brain Science, which relates to the creation of an adult human brain atlas. So we've also done a developing human brain atlas that I'm not going to talk about, because for the purpose of HubMap, really just here, adult, we have our hands full for thinking about cell types. I'm also not even going to show you the table, the ASCT table, because it's actually disappeared. So it was in an earlier version, but it's not in the 10 by 10 map that we have right now. So I just wanted to let you know that there is a spatial anatomical reference space that parcelates annotated structures with a central coordination framework. And all of these are hyperlinks here. So I encourage people who are interested on their own time to follow up through this PowerPoint presentation or the presentation here, because each one of these links to the access for these sites. So the newest 3D version for the brain is available through the link shown there. There's 141 annotated structural brain areas that also have 3D meshes that are downloadable and can be viewable through different visualization platforms. This right now, in terms of developing the anatomical reference, is on hold at the Allen Institute for Brain Science. So this is basically a fairly static definition of these anatomical structures. But the real question we want to get to is, what are the cell types in the brain? And I have said this many times to Kati, and she keeps asking for more, but we don't know. So the entire purpose of the Brain Initiative is to define those cell types. And
Amy, she does the same thing to all of us, so she's not picking on you.
No, I know, it's fine.
And the answer is always the same. We don't know. That's why we are doing this.
Yes. but it's a really broad problem. I mean, we have things like neurons and non-neurons, and one of the challenges is that we don't yet have those anatomical definitions for the whole brain. So what we do have is we do actually have the motor cortex where we have those cell type definitions. And I just have linked out the most recent paper that defines those here. There's also a fully sort of described set of ontological frameworks that has been maintained and is continuing to be updated through people in Uberon, specifically Chris and David. So I think some of them might be on the call, I'm not sure.
Yeah.
But this is in collaboration specifically with the Brain Cell Census Working Group that's doing that. So I'm leaving it to them to talk about that. Okay, so maybe we could just move to the next slide quickly. I don't want to take too much time on this because I know we have a lot to get through. So lessons learned, or lessons in progress, I guess I should say, is that the Human Brain Atlas right now is at the gross anatomical level. And then the next step will be to map in overlapping cell types through doing the kind of molecular analysis that's based on single cell analysis that's very much ongoing in the Brain Cell Census Network projects. So that has already been done for the motor cortex, or M1, and that is referenced on the previous page just in terms of what that publication is, and all of that is contained within essentially not just within the publication, but within the set of cells that are being included in the cell census for human brain. So the entire project is working on this, and this is spanning out for the next 3 to 5 years. So this is very much of an ongoing process. So really for right now, what we can do is use this anatomical framework. Katie already has this essentially pulled in and is very up to date, is working with James G and others to make sure that, that there's cross-referencing for HubMap, which is key. And it's really just about iterating when we start getting those cell types, mapping them into that central framework, which we're positioned to do. So using those anatomical, both ontological as well as spatial reference frameworks, that's where the iteration is going to be. And so right now that is available for the motor cortex, but not for any other brain structures, 0. So we can do things like refer to ontological frameworks that call out different substructures and have classes of cells that are described not using molecular markers, but none of those are mapped into this central framework. So since we're doing this for brain anyway, for human brain, I would just recommend, essentially rolling it into when that data is available. But that requires patience. So with that, I just wanted to also call out 2 things. So there's a preprint that we have now in archive from the Allen Institute for Brain Science, where we've just described a framework that we're using for nomenclature of cell types as it relates to connecting it with a taxonomy. So I've linked that here. I encourage you to take a look at it. It's under, we've just submitted it to Eli for review, so it's not formally out there, but it's also on our Allen Institute website that just describes the convention. But basically it's proposing that for anything that you have genetic cell type based transcriptomic information, that you use that taxonomical information to link to specifically what the nomenclature will be. And this is specifically for the purpose of allowing us to sort of reference across the whole body, ultimately, what those, what the nomenclature of different cell types might be. So it's kind of a big deal. It's just a starting point, but at some point, we do need to think about what are the nomenclature conventions that will allow us to reference information across cell types for the whole body. So for example, if we have, thinking about blood cells, if we have vasculature and cells that are part of the vasculature in the brain, what is the framework that we're going to use to have shared nomenclature across the vasculature framework and then across the presence of the vasculature in the brain? And you can think that for any organ system. So this is just a proposal for a convention to use for that. So for people who care about nomenclature conventions, would love your feedback on that. So that's pretty much it. That's, I think, my 7 minutes. And any questions or comments are welcome.
Thank you, Amy. Are there questions, comments for Amy?
Amy, this is Todd Valerius. Just a quick question. With the molecular underpinnings for some of the, as new cell types emerge, does the proposed nomenclature flexible for taking on new ideas? Yeah, that's the idea.
Absolutely, that's the idea. So the idea is that anytime you would have a anytime you would have a cell type, you would need to essentially co-present it with some form of a taxonomy, a reference taxonomy. So the concept is that these things go together. And it's a little bit kludgy in terms of how operationally it could work in the future, which is of course always the challenge. But it's sort of thinking more about the way in which reference genome information was originally used, where you have a gene ID, but that gene ID essentially links back to the concept of a reference genome. So you think about, you'd have a cell type ID, and that would also then call back a reference into a taxonomy of, that would provide provenance into how the data was generated, how that data was normalized, what that data set contains, and then that would essentially be a versioning mechanism. So it's not trivial, it's conceptually simple, but operationally it will require work. And that's actually one of the reasons why we're trying to now, socialize this and get this going. And I'll cross-post it where I need it to go. But yeah, I would encourage anybody who is thinking about an extensible convention of nomenclature to at least look at this and then provide feedback of, well, can this work? Does this work? What doesn't work about it? So that it can adapt. It'll be a work in progress.
And I think that would be a great topic for one of the future meetings we could have together, because I think we all are trying to understand how to use existing nomenclatures, what's working, what's not working. And actually the 3 subsequent brief presentations, which were supposed to be 7 minutes, but maybe now have to be 5 minutes, so that we can all fit them today, are helping us all understand a little bit more about the existing cell ontologies, standardization of gene names, but also what are the issues if you now try to take those ASCT tables and try to flatten them. So again, in the interest of time, I would love to have Anna Maria Massi present maybe just 5 minutes, if I could ask you that, Anna Maria, because I would love to see what you can help us all understand. And again, we all come from very different points of view here.
Okay, we'll try to be very quick. And at least I was just starting with the history of the cell ontology, because to understand why the cell ontology is structured like it is right now, and then how it's going to evolve to be adapted to different type of data. So the cell ontology, and was starting in 2005 and was by Bard, but had a problem because the cell were defined by using different methods, so by organism histology, functional, and that was very confusing, because you will not have any single parent for each single cell type, so it was very confusing. So next slide. In 2009, when I started working in the ontology, together with Lindsay Cowell, we decided to use one cell type, in that case was a dendritic cell, to kind of see if we could change the approach that then could be used to modify and update all the cell ontology. And so that's what we really did. And that's type of format of the cell ontology, that is what you see at the moment, came mostly from my background, because I was coming from a lab and was working with the cell, and I was thinking the way to define cell without looking at, because when you look like in the histology, you can have information just looking at, you can discriminate, but the computer could not know. So it was the fact that there was available the FACS method, so the cytoplometry, and in particular you could identify cell by using cell marker. And so we decided to take that approach and start to define cell by their marker that they were expressed. And here is just a kind of, how it is right structure. And so we have cell defined by the marker they express, cell as particular function, like a, I don't know, kinase function, they can have a disposition to do something. So the ability to do something like proliferate. And they develop from other cell type, and they transform of, and this in particular, the transformation of, was referred to cell that pass from immature stage to mature stage. And so we could capture that link between one cell, immature cell to the mature cell, and then where they were located. In this case, you see that we are now using the part of most of the time. But in this case was location of, because I was talking about the hematopoietic cell type, And so usually, in particular, the these cells, they migrate through the body and are not like macrophages that you can find in the tissue. So they are tissue resident macrophages. These were moving, and the relation was more location of instead of autonomy location, because they are not always be in one specific organ, but they can go through different organs. And so this was mainly the approach that we took at the beginning and is still now on. But obviously, this now cannot really, can fit in part with molecular data, especially with single cell data, but still I think now the cell ontology is trying to look forward to check how to integrate that part to this structure. And obviously has been, if you go to the next slide, has been developed, all the other cell type, taking as example, the dendritic cell ontology, and Alexander Deal with Chris and with David, they have done a lot of work on that. And in particular, one of the last, and this is the last paper from David, where he's really trying to make the point that how we should get the cell ontology, going forward in order to be driven by the data, and this time in particular by the single cell type of data. So how can integrate that to whatever we have there? So linking the expression of the RNA with the protein or the cell marker that we have now. And that is work in progress, and we are discussing, and so, but that's how we are going to do as a cell ontology. Then I want just to go, if you go, yes, on the next one, and I'm trying to be quick. So I was looking through the table that Ellen has done, and I was capturing some of the things that she has, like cell not found, or some of the issues that she has, maybe because this could be useful to us to kind of approach differently. So in this first case, I don't know if you can clearly see in my slide where it's seen better, but under the cell type column, we have, for example, vascular endothelium, and then we have macrophages as light cells, if I'm not wrong. And obviously, this is something that we would like not to have, because the vascular endothelium is not a cell type. And so we should have, I think, another column, either to have this under the structure, or what I think could be more correct, have another column where we can list the tissue, and then we have a cell type that are, part of that tissue. And so this is one of the things that I noticed. And then if you go on the next one here, and is the other, these are just example of what, there are more than one, obviously. So cell type that are not found. And then in that case, the point is that we need to ask if really is a new cell type, or if it's maybe they are just, is the naming different, we cannot find it. And so the main point is how we can figure out if it's the same cell type, if it's a new cell type. In this case, I think it's a new cell type, but since in the ontology hierarchy, any characteristic of the parent is inherited to the children, in order to be sure that this is a subclass, for example, this is a reticular myofibroblast, and if it's a subclass of the general myofibroblast, we should go and look at the definition in the cell ontology. And if the definition, like in this case, as SMA positive, CD34 negative, and CD45 negative, fits with the cell as a reticular myofibroblast, then can be considered a children. And in addition to that, we can then add to this new cell type additional markers that will be specific for the new cell type that are not going to be present in the parent cell type. And then the last, I don't know how many minutes I have. I think I have the other slide. And another issue was general cell type versus specific cell type. Because again, in this table, I found that they were listed T cell type, and they were all referring to the same cell ontology class, general class of T cell type. But looking at the marker, you could see that they were two different types, because one was CD4 and the other one was CD8. And so I think if you can go to the next one, the point is that we want to be, I think, as much as specific as we can when we know the information, because you know, in the cell ontology, this is a T cell, you have all this region, then you go to the CD4, CD8, for example. So if we know if we don't know the information, it's good to mention and list it as a general cell type. But if we have a more specific information, I think we should always try to keep a link to the more specific term in the cell ontology. And I think I have done, I don't know if I've done in 5 minutes, but this is just to, you know, have a little bit of background and example.
Thank you, Anna Maria. This was excellent. And it's also great that you used examples from the existing ASCT tables, which of course I also care deeply about. I also know we have Chris Mungle here on the call, which is really wonderful to have you as ontology experts, because we will need you. We will need your expertise and we want to benefit from it. So let's see if there are quick comments and questions. There's some relevant information in the chat window also for those of you which want to check that out.
Hi, this is Kathy Reinhold from the Broad. We're trying to map single cell data, and I'm very excited to hear about the cell ontology normalizing definitions. So would love to chat.
Yeah, that's you know, it's not going to be, you know, straightforward, because there is obviously the single cell is talking about expression level, and sometimes the expression, the mRNA can never be, you know, translated into protein. So, but we need to, we are discussing a way of having a kind of cell type that is kind of capture the expression data, and then link somewhere back to the protein expression level.
And we've also been tracking sort of an a priori cell type and then an inferred cell type from analysis.
Oh, that's good.
Very good. All right, in the interest of time again, I think we have time for one more agenda item. So Alan and I will probably have to hold our horses to the time in a month, which is not what we planned to use or to do. But I think that's the best decision. And we have Gokkin and Francois presenting on standardization of gene names and genome builds. For those of you which have to run off earlier, the next meeting will be on August 6th. There will be an agenda shared soon. And then you can always suggest agenda items. Please do so. Now over to Gokkin and Francois.
Hi, can you hear me?
Yes. Mm-hmm.
Hi, my name is Gökçen. So thank you for the invitation. I'm a postdoc in Avira Gev's lab, but Kristin, Francois, and I are like collaborating in a single nucleus atlas project. So we had a few like brainstorming sessions about the marker tables, and which we also used in our project. And then we just decided to like give some feedback. So this is a nice cartoon that I would like to show in the standardization meetings. It should be shown maybe in all the meetings. So it basically says like there are 14 existing standards, and then with like one complaint, like we need like a universal standard, and then the number of standards becomes 15, something to maybe keep in mind. But the main feedback is about the gene name or ID or protein name. So we have these marker tables or different resources which list like different markers for different cell types and so on. But the name ID or protein name is an important decision, and I think the important thing is maybe it depends on who the users are. So in our case, we are using it for single cell data set, and human readable gene names or IDs are these are the things that we are using. But they are both subject to change. So gene names evolve, gene IDs also evolve, but IDs are like a bit more reliable. Yeah, but even with different versions of the same resource, they might also change like between ensemble ATT 100 version, for example, some IDs are removed, some new IDs are added. So it doesn't fully solve the problem, but it makes the resource a little bit easier to use at least. And on the other hand, the standardization within a project is easier. Like if you make a marker table, for example, and if you put also which gene names and IDs you use and in which version, then it will be easier to use for like other people. Yeah, Francois, are you also here? Do you want to talk about the, yeah.
So along the same lines, essentially in terms when we map reads against the reference genome for quantification, the choice of the reference used can have a large impact on quantification. Perhaps not globally, but for certain genes, that's definitely the case. If you're curious for an example, one where we saw massive differences just recently is a very well-known gene, transferrin, where the UTRs have completely changed between recent versions of GenCode. And so that's for 3' based quantification methods can have a large impact, for example. And so essentially knowing when we have marker genes, sort of knowing what reference annotations went into actually quantifying their expression can be quite critical. And similarly to some degree when we're dealing with single nucleus data, where reads can both be intronic from pre-mRNA and spliced, the way the reference annotation is built for the mapping can also have quite a large impact. So essentially not having just a marker, but also some kind of history of how expression for that marker gene was quantified can be important in some cases.
Mm-hmm. So the next slide, I think we can switch to the next slide. Yeah, so on the right side, we put like a screenshot of the marker tables that we generated for a specific Atlas project. It's a very simple thing. It's not as advanced as the ASCT, but we just see like the gene names, unlike what I suggested, basically using gene IDs, but just for the sake of simplicity and convenience, we use the gene names and like equivalent, like homolog mouse gene names and some like compartment information, cell type, and the link to the like the citations of the markers. So yeah, I think keeping citations also makes sense. This was something we wrote in the email. And another thing is like for the projects with like multiple tissues, like having the distinction of like the pen markers and tissue specific markers is also important. Like markers that are the same across all the tissues, more canonical markers, or more tissue specific macrophage markers, for example, or fibroblast markers. So specifying this was also essential. And other than that, yeah, I think maybe you might have discussed that already in these meetings, but like some cell types are like harder to define, like more fluid ones, for example, like keratinocytes that look more like a continuum rather than like a discrete type. These are also problematic. And the last thing I would like to maybe emphasize is the agreement between the cell types. So like in fibroblast 1, fibroblast 2, or like TUFT1, TUFT2 kind of naming and standardizing these names. This is also a very important thing. Yeah. And in the next slide, this is the next one is, yeah, this is the last slide. So like a few more details about like how the markers are used. I think this is also important. Some people use it just like in single cell, just for scoring, using these set of genes and then looking at like the average expression and then setting a cutoff and then annotating these cells as like cell type A or cell type B. But it's not as easy always. So the semantics, the meanings of the markers might also be different. So I have an example here, for example, like positive versus high. So we have some cell populations. It doesn't really matter which cell types these are, but like MGP high, for example, can be used to identify like these 3 clusters that you see in the MGP panel. So this is like high kind of marker, but like in some cases we have a lot more specificity and we can use just like post and positive, for example. So putting just the gene names there or gene IDs doesn't really fully solve the problem, but it also makes sense to explain somehow or communicate how these markers will be used. And the last thing is about the specificity. So I was thinking something like a GWAS catalog, like instead of putting only the markers, adding more statistics about these markers, like how specific it was in dataset 1, dataset 2, and dataset 3, and like some p-values or enrichment scores or specificity quality scores, for example. Because not all markers are like of the same quality. Some of the markers that we see in some papers are not useful to us at all for some reason. Maybe the technologies are different or granularity is different somehow. But some markers are like a lot more specific than the others. And the very last bit I would like to talk about is the context. So here I have an example of live1, for example, in the right panel. So there are like live1 positive macrophages, which is like the right island, upper right island. So like within macrophages, we can use live1 as a population, like a subpopulation marker. But if you look at the entire data set, live1 is also expressed in the lymphatic endothelial cells or even some myocytes, for example. So it's the marker, the concept of markers are also like context dependent. So if you use live1 in the entire data set as a macrophage marker, that would be like super misleading because it will also mark the lymphatic endothelia as well. So context dependence or context by itself, I think also matters. So adding some kind of statistics or specificity scores in the marker tables, I think that would also be a nice idea. And I don't know if you discussed, but like negative markers are also used sometimes in the literature. Yeah, so that's all we have.
Thank you so much for presenting that work. And it's great to see actually your version of the ASCT tables. I know you developed those independently. And again, I love to see the citation references in there. Amy Bernard from Allen Institute, she also posted into the chat log before she had to leave, or she's still with us. Do you want to, I think you have to run, but if you want to comment on it also, feel free.
Oh, I have to run in a minute. I love this. I think that this is one of the things we were trying to address again with that preprint that I just posted here. It does still beg the challenge for all of us that we need some form of a central repository, sort of a management framework that kind of goes maybe a level above GitHub or individual references for this. And that's actually one of the things we propose a need for in this article. But yeah, I love particularly the whole purpose of saying that just the gene name and ID is not enough, moving on to that next level where it infers that there does need to be a structural reference framework. And I don't think that we have that at the level of cells. We do have it at the level of genes, but we need to bring it up for cells.
Mm-hmm. Very good. I know many of you have other meetings waiting. I will stay on for a little bit, but thank you all for presenting today. This was excellent. There are more slides, as you might have seen, and so Ellen and I will present next time when we all see each other in a month. Until then, stay safe. Have a wonderful 4th of July. Stay in distance if you can. I don't want any casualties in this team, and just come back. And also propose agenda items. I welcome that. Thank you. Bye.